So did you get a message that we are going to record? Yes, okay, good. The next question, I would like to know you a little bit, so tell us where you come from. Oh, that's interesting. Last year, when I taught this course, we had people from all the departments. Seems you are all from physics. Anybody not from physics? Okay. Not everybody answered. There is a question here about next week. This is the election day. Yes, we will meet next week. And the last question for this batch is, what do you expect to get out of this workshop? You can use 3 words, just punch in whatever you want. Well, very nice expectations. What we are mostly going to do this course is to learn how not to do bad science. And you will see, I hope, that during these 13 meetings that we are going to have, is that there are lots of wrong things right now, the way we do science. All right, so let's move on. What we are going to cover during this course is today, and the next appointment is going to be about what is a scientific method, what is a peer review, replication crisis, and I think we might be able to surprise you here. Then we'll discuss open science and beyond. So that's going to take us about 2 weeks. Then we'll cover how to write a good paper, how to critically read a good paper. We will use the famous Watson and Crick from 1953 as an example for a good paper. We'll then move on to my own research area, and we'll talk about Forster paper and about fluorescence resonance energy transfer. You will get a home assignment about a paper that relates to this topic. This is a topic that most of you are not familiar with, and will basically ask you to read a paper and see what you understand reading a paper not from your field. And then we'll cover scientific fraud and misconduct. That will take us 2 weeks. If we have time, we'll go over the home assignment that we gave you here and basically go over the answers for the questions. And from this point on, the rest of the course is actually going to be given by you. Any questions so far? You can open your mics and any comments. If no comments, let's move on. The way we are going to grade your work here, participation is 15%. The home assignment, the questions that we are going to give based on a paper on single molecule threat, is going to be 20%. You will have to write a 2 pages summary of a paper of choice that you're going to also present as a PowerPoint for the whole group in English. So you're going to send us both a PDF, 2-page PDF, and you're going to present a PowerPoint. PowerPoint presentation will be around 25, 30 minutes, 20 minutes presentation and 5 minutes questions, something like that. You will have to consult with me about what paper you're going to present, paper or a group of papers. I specifically did not put dates here. This will come as we go and we'll adjust dates accordingly. So the main assignment, as I said, you're going to analyze a paper, you're going to write reports and present a PowerPoint presentation on this paper analysis. And just a reference for how to write a peer review paper is given in this link. This could aid you in preparing your own presentation and PDF summary. Regarding the PowerPoint presentation, what we expect really is for you to tell us a very good story. Misconducts are always making very good stories, but sometimes very good papers also make good stories. So we'll talk a lot about this as we go along, but we expect you to pick up really interesting stories to tell the whole group. And I'm going to give you some really, really interesting stories. So as I said, exceptionally good paper can also be a good story. All right, so let's move to what we are going to cover today. We are going to discuss the scientific method, the peer review, the failure of the scientific method, mostly due to the replication crisis. And I hope that by the end of next week, you'll be convinced that there are some big issues in the way we conduct science nowadays. We'll discuss the remedy that the community is coming up with, which is going under the umbrella of what is now called open science. And we'll discuss what could come beyond open science. So let's discuss the scientific method. So tell us in your own words, what do you think constitutes a scientific method?
Actually, I didn't really understand the terminology of scientific method. What do you mean?
A process of confirmation.
Mm-hmm.
questions, data, conclusion, experiments. Exploring. Guys, this course is about participation. I do expect you to participate. So 4 responses are not enough. All right. Statistical proofs, specific way of science, broad agreement in academy. Anything else? All right. So this is taken from Wikipedia. Let me read it to you. The scientific method is an empirical method of acquiring knowledge that has characterized the development of science since at least the 17th century. It involves careful observation, applying rigorous skepticism about what is observed, given that cognitive assumptions can distort how one interprets the observation. It involves formulating hypotheses via induction based on such observations, experimental and measurement-based testing of deductions drawn from the hypothesis, and refinement or elimination of the hypothesis based on the experimental finding. These are principles of the scientific method, as distinguished from a definitive series of steps applicable to all scientific enterprise. So it involves careful observation, formulating an hypothesis, running experiments, and deducing conclusion from experiments based on the hypothesis and the questions we asked, and then refinement by elimination or by confirmation based on the finding. The scientific method is the process by which science is carried out. As in other areas of inquiry, science through the scientific method can build on previous knowledge and develop a more sophisticated understanding of its topics of study over time. Basically, we are moving the front of knowledge forward. So let's go through the steps of the scientific process. We have to formulate a question based on previous knowledge, put forward a new question, a new hypothesis, a prediction. Then we are going to test this prediction with an experiment or with some simulation and so on. We are going to analyze our data, deduce some conclusions. We are going to replicate and retest, and we will see that here we have a big failure nowadays. We will basically write our results, submit it to other people to basically decide if our understanding and conclusions are correct or not. This is called the peer review. We are going to record and share the data. We are going to publish, and then we are going to ask a new question based on how much we moved the knowledge front forward. And this is basically an iterative process where we ask a question, answer that question, and therefore ask a new question, and keep on doing this. So that's basically the steps. So let's analyze the case of Watson and Crick. We are going to come back to this when we analyze how to write a good paper, but let's just see how the discovery of the structure of double-strand DNA as a double helix was constructed in terms of the scientific method. The history of the discovery of the structure of DNA is a classical example of elements of the scientific method. in the early 1950s, it was known that genetic inheritance had a mathematical description, starting with the studies of Mendel, and the DNA contained genetic information. This is Oswald Avery's experiments and the transforming principle showing that basically DNA carries the genetic information. But the mechanism of storing genetic information as genes in DNA was unclear. Researchers at the Bragg laboratory in Cambridge University made X-ray diffraction pictures of various molecules, initially proteins, starting with crystals of salts and proceeding to more complicated substances. So using clues painstakingly assembled over decades, beginning with the chemical composition, it was decided that it should be possible to characterize the physical structure of DNA, and X-ray images would be the vehicle. So basically, in the early 50s, they already understand that in order to decipher what is the structure of DNA, the best method to do this will be crystallography. And one needs to purify DNA and somehow crystallize it into a crystal structure that can diffract. And if you can get diffraction from DNA, you could solve the structure. So all of that was known. So here comes the hypothesis. The first hypothesis was wrong. Pauling, who was at Caltech in California, proposed that DNA might be a triple helix. This hypothesis was also considered by Francis Crick and Jim Watson, but they basically discarded this. They said that this doesn't make sense. There are some contradictions. So when Watson and Crick learned of Pauling hypothesis, they understood from existing data that Pauling was wrong, and that Pauling would soon admit his difficulties with that structure. So basically, there was one hypothesis. Based on whatever they knew, they decided that whatever Pauling is thinking is wrong, and they came up with a different hypothesis. So the race was on to figure out the correct structure, except that Pauling did not realize at that time that he was in a race. We'll talk about this later, but anybody knows why Pauling kind of missed this? It's a good story. If you know it, just turn on your mic and tell us. Nobody knows. Okay, so Pauling was a pacifist, and we are talking about the time that in the US, McCarthy was basically prosecuting and going after communists and pacifists. This is after the Second War and just before Korea. I'm sure you heard about the term McCarthyism. So Pauling was a victim of McCarthyism. Because of his political views, he was banned from actually leaving the US and traveling for conferences. There was a conference in, I think, 1952 in Europe, where the data about the diffraction pattern of the initial results of diffraction pattern of DNA came out. And if he was able to go to that conference, he probably would have gotten the structure before Watson and Crick. He was not allowed for political reasons. So that resonates very well in our days because we see a lot of this happening today as well. All right, so Watson and Crick and others hypothesized that DNA had a helical structure rather than a triple helix. They basically hypothesized a double helix. This implied the DNA X-ray diffraction pattern would be in X shape. Basically, they have done some calculation. You know that Bragg diffraction relates to interference, diffraction from different planes of the crystal. And based on the hypothesis that DNA is a double helix, they've done mathematical calculation predicting that the diffraction pattern of a double helix in a crystal should have something that looks like X-shaped on the photographic plates. If it's not X, it will be a different structure. But if it's X, that indicates that it's a double helix. This prediction followed from the work of Korshan, Crick, and Band. That was a mathematical, at that point, they did not do simulation. Everything was analytical by hand. So this prediction provided mathematical explanation for the empirical observation that diffraction from helical structure produced an X-shaped pattern. And in the first paper, Watson and Crick also noted that the double helix structure they proposed provide a simple mechanism for DNA replication. So beyond just solving the structure, they already proposed in that paper, a very famous paper, and we are going to go over this paper in detail. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. At that point, this was still a big puzzle. How is the genetic information being propagated when cells divide? And just based on the structure, they were able, the hypothesized structure, they were able and suggest a mechanism how DNA is being replicated. This is considered to be one of the biggest understatements in a scientific paper. They are being very modest here in this sentence, are being very careful, but obviously this particular deduction started the whole field of molecular biology. So Watson and Crick showed an initial and incorrect proposal for the structure of DNA to a team from King's College. They were in Cambridge, but in London, in King's College, there was a group, Wilkins group, where Rosalind Franklin was working together with, and Gosling. Franklin immediately spotted the flaws which concerned the water content. So basically, the first hypothesis by Watson and Crick was wrong. They were meeting and talking to this group in London. Rosalind Franklin, who actually did the experiments, pointed out that their first hypothesis is wrong. And later, Watson saw Franklin's detailed X-ray diffraction image. Sure enough, that image showed an X shape, and we'll see this in a second. And they already knew that if it's an X shape, it's going to be a double helix. Here, there is one of the biggest cases. I don't know if we can call it misconduct or not. We'll discuss this. Anybody knows what is the story of Rosalind Franklin? If yes, please turn on the mic. Who took the data for this very famous paper?
I know that was.
I cannot hear you. Just a minute. Something
I know
is
that
wrong. Just a minute.
now you're in me.
Can you speak again?
No, now it's okay. It's
Yeah.
good.
Yes.
I know there was some woman that took the data, and a few male scientists use it. Use their data.
Yeah. So Rosalind Franklin is the lady who actually took the data that constituted the Nobel Prize. Her advisor showed her data without her permission or knowledge to a competing group, to Watson and Crick. Based on the data, they showed the rush to publish a paper without acknowledging her. And based on that, they got the Nobel Prize. Unfortunately, she died very young. And after that, I think that was tuberculosis or something. Very, very sad story of almost scientific misconduct. Some people think it is pure misconduct, but this is a story. We'll talk about this more later when we go over the paper. But what I wanted to show you here is just how the front of knowledge is progressing based on the scientific methods. So there was some hypothesis, experiments, calculations, confirmation, new questions, and so on. Here is a famous photo 51. It's called photo 51 because they tabulated all the data, and here you see that the diffraction pattern has that X. When Watson saw that X, he knew they solved the structure because their initial mathematical calculation predicted that structure. So basically, based on this, what Watson and Crick did, they went to the lab, they cut pieces from cardboard, and they built a model for the double helix. And there is a famous picture of Watson and Crick on, let me see if I can quickly show this. So we'll talk more about this, but this is a highly recommended book. It's showing actually you see the mirror image, right? So this is a Watson book, The Double Helix, highly recommended. So basically they build a model for the correct structure using this information along with previously known information about DNA composition, chart graph rules of base pairing. Basically, there was some chemical information which basically people knew that you have the same amount of thymine and adenine, or those are T's and A's in the DNA, or the C's and G's. And based on that, and based on the X-ray structure, they built a model and quickly rushed to publish a one-page paper that got them the Nobel Prize. After considerable fruitless experimentation being discovered by their superior and being discouraged by their superior from continuing. Basically, the boss of Watson and Crick told them at some point, stop. This is not a productive area of research. That did not deter them. They continued. Numerous false starts. Watson and Crick were able to infer essential structure of DNA by concrete modeling of the physical shapes of the nucleotides which comprise it. They were guided by the bond lengths, which had been deduced by Linus Pauling and by Rosalind Franklin X-ray diffraction images. And a new field of molecular biology was born. All right, so that was the scientific method. And as I said, we'll look into this paper much more carefully in 2 weeks from now. And the next topic is the peer review. This is actually a very interesting topic because we'll talk about how it was done until now, and we'll talk about some changes as recent as a paper from 2 days ago. And this is really a response to the failure of the scientific method. We talked about the scientific method. We are trained to think that we are all scientists doing the right thing, we have the right method, and science is doing great. In this course, we're going to see what are the problems. And we are facing a huge problem nowadays, and the peer review is changing because of that. All right, so again, we are referring to Wikipedia. Let's discuss what peer review is. Or referencing of papers. It's a process of subjecting an author's scholarly work, research, or ideas as a proposal to the scrutiny of others who are experts in the same field before a paper describing this work is published in a journal, conference proceeding, or a book, or funding agencies decide to fund the proposed work. So why peer review? Why do we need peer review? An external point of view. Objectivity, because nobody is perfect. Is the peer review make our science better? To question our findings? get objective evaluation. So this is a repeat of the same idea.
Hello.
Yes.
Can I speak? He means because I'm not connected to Mentimeter yet. So
Sure,
so
of
can
course.
I?
Go ahead.
Okay. Yeah, because well, if we try to propose some kind of ideas or we did some kind of research, so we need some external point of view in the sense that whatever we did, some other expert committee will come to, check that things whether we are the right pathway or not. Because well, there can be a problem with what we did because
So basically,
it depends upon.
yeah. So the external people who are more objective, who can kind of
Exactly.
correct
Yeah.
us and find mistakes and so on.
Yeah.
All right. Anything else? All right. The peer review helps the publisher, editor-in-chief, or program committee or funding agencies to decide whether the work should be accepted, considered acceptable with revisions, or rejected, funded or not funded. Peer review is generally considered necessary to academic quality. So we are all used to think that the peer review is really what keeps us in check and makes science correct. However, peer review does not prevent publications of invalid research. The fact is we have lots of retracted papers where the conclusions were wrong, where there were problems with experiments, or even worse, when misconduct was conducted and then papers are retracted. And somehow peer review misses all of this. In fact, nowadays, there is a field of science called meta-science. We'll talk about this new field, which is using scientific methods to study science itself. And there are quite a bit of evidence, in fact, lack of evidence, showing that peer review improves the quality of published papers. So there is no evidence that the peer review is helping in any way. That's some food for thought. We are used to think that the peer review is really the ultimate, but it is not. And I'll try to convince you in this. So what is the justification for peer review? External reviewers with special expertise can spot mistakes or omissions. Sometimes they can, sometimes they miss. Reviewers are often anonymous and independent. There are different styles and types of peer review. You can have either single or double-blinded. Single means the reviewer knows the names of the authors don't know the names of the reviewers. Double-blinded is that neither know the names of the other side. What is the procedure? So if you send a paper for publication, you first send it to the editorial board of the journal you're interested in, and it undergoes some kind of what is called desk evaluation. So the editor is quickly looking just at the title and abstract and decides if this is of interest to the journal or not. If it is, it will be sent to reviewers, and it's usually going to be blind review with respect to the reviewers, so the authors don't know who reviewed the paper. The reviewers will ask for revisions, most often, and the authors will then basically fix the paper and send it again. So as we said, there are several different types of peer review. There is anonymous or attributed. There is double-blinded. There is an open peer review, which is becoming more and more popular. Basically, we put the paper and the review online, and everybody can read and discuss this and start a blog and have some iterations. We can have a review which is called a pre-publication review, where people basically register the paper, what they plan to put in the paper. It undergoes some kind of review, but once everything is ready, it doesn't go through another review. Or a post-publication, which is mostly open peer review, where after the paper is published, you start to write comments on some kind of a blog. And you can have an informal peer review, which is basically Twitter or similar. So here is an example of editorial in Nature for March of this year, which talks about trial of transparent peer review yields promising results. Because of the crisis that we'll discuss in a second, there is a big movement toward changing the peer review. Here is an attempt in Nature where they are making the reviews publicly open. They publish the reviews as well. And what they say here, making reviewers' work public illustrates the lengths that researchers will go into the service of scholarship. Trial transparent peer review yields promising results. So basically, they hail this idea of openly publishing the reviews together with the papers. This is from February of this year. Nature Method comes with a new way of doing peer review, and they call it registered reports. Nature Method is introducing a new article format, register report. We encourage all authors interested in submitting comparative analysis of the performance of established related tools of methods to familiarize themselves with this alternative approach to peer review. So basically, you are submitting a paper which says what you're going to do, and that's going to be reviewed. But once you are finished with your paper, it doesn't go through a review. This is called a registered report. And this is from 2 days ago. Peer review without gatekeeping. So this is eLife. eLife was one of the very first open science journals, starting around 2010 or 8, I don't remember exactly. This was started by a group of professors in Berkeley who really did not like what Science and Nature are doing, where all the decisions about what is published and not what is not published is really somehow affected by some commercial interests, and this contaminates the whole idea of how science should be done. So they were pioneering in this idea of open publications. Nowadays, almost all journals have a version of open publications following eLife. And here, as recent as 2 days ago, they are coming with this idea of peer review without gatekeeping. eLife is changing its editorial process to emphasize public reviewers and assessments of preprints by eliminating accept-reject decision after peer review. They are still going to decide if the paper is good or not good for eLife. If it's not good for eLife, it's not going to. But if the editorial board decides that the paper is worthy of a review, they will publish the paper and they will publish the review. But they will not tell you if the paper is accepted or not. This is really up to us to decide, because once you publish the review, you can comment on the review, and over time, there will be enough responses, and eventually the community will decide if the paper is significant and the results are important. So this is a big change, and we should all watch out for how other journals are responding to what eLife is doing here. We have found that these public preprints, reviews, and assessments are far more effective than binary accept or reject decision ever could be at conveying the thinking of our reviewers and editors and capturing the nuanced, multidimensional, and often ambiguous nature of peer review. eLife will now let them stand on their own by publishing every paper we review, along with our reviews and an assessment as a reviewed preprint, a new type of research output we hope will become the norm across science. Again, this is a big change, and it's a response to the replication crisis, and basically follow up on the idea of open science. More significantly, the emphasis placed on directing papers into journals has turned journal names into the de facto currency of academic research careers and institutionalized the practice of judging scientists based on where rather than what they publish. This has in turn transformed journals from means of communicating science into gatekeepers whose judgment, ones that are heavily influenced by bias, fetishness, and chance, can determine which science gets seen and which scientists succeed. So basically, the biggest criticism for the peer review is that if you have not published in Science or Nature, you should not get tenure position, or you should not be promoted from, associate professor to a full professor. You don't have enough papers in Science and Nature. But Science and Nature decisions are not purely science decisions. These are commercial entities. They want to sell their journal. So somehow they got to become the gatekeepers. They make decisions, what is good science, what is not good science. People's careers are dependent on this. And it's somehow contaminated and tainted by commercial interests. So the question is, can we do something better? And here eLife says, we want to do something better. We are not going to decide what is good and bad. You as the community are going to decide what good as bad. If the paper is of interest to our editors, we are going to publish it, and we are going to publish the review. And then we open the discussion and other people can comment on the review.
Can I ask a question?
Yeah, just a minute. Let me just finish this page.
Okay.
Authors should be able to share their work freely and openly when they think it is ready. Peer review should consist of scientists publicly sharing their assessments of already published papers, either under the auspices of the editorial organization that oversees the review process or on their own. Works of science should be reviewed by multiple relevant groups and individuals throughout their useful lifespan. So basically, the old way of doing peer review, where 2 or 3 reviews are assigned by an editor, they are supposed to be experts. Nowadays, most established scientists are so busy writing grants, writing papers, doing a lot of administrative work. Usually we'll say, well, we don't have time to review a paper. Let's give it to our postdoc or to a senior student who is going to be reviewing the paper. So lots of decisions, peer review decisions, are actually made by young, inexperienced scientists. And eventually that is impacting careers of people and promotions of people based on not so good peer reviews. So the idea here is to open it up and have a response of the whole community rather than rely on 2 or 3 reviewers. And officially there might be established scientists, but it could be the students who are doing the review. There was a question. Yes, go ahead.
Yeah, actually, I was about to ask, like, if you said that what eLife is trying to do is if I write a paper, I'll put it in a kind of a open source kind of a web page or something. And the scientific community will decide whether what things are right or not and things like that.
No, not exactly. So you are kind of mixing between repositories and journals.
Okay.
So we'll talk open science. Nowadays, the practice is that before you submit your paper to a real journal, you first put it on archive, on bioarchive. Those are depositories. And yes, people can respond to whatever is shown in the depositories. But here we are talking a real journal. There is a real journal with real published paper, which went through the editorial board. Okay. The editorial board decides we are going to review this paper or not. Once there is a decision to review the paper, it's kind of accepted. It's accepted in the sense that the journal is going to publish this paper and publish the reviews.
Yeah.
And then open up a blog where other people can respond both to the paper and to the reviews.
Okay.
The author's names will be there, the reviewer's name will be there. So the reviewers are basically signing, yeah, this is what we agree and
Okay.
what. So they are exposed.
Okay. Okay.
If
That's-
later on, there is some criticism about the reviewers because they kind of missed something or screwed up, that will be discovered and, people will comment on this. So there is vulnerability to the reviewers as well. Until now, reviewers were blinded. You did not know who the reviewers are. And you'll have to take, the editors will say, these are the reviewers, that's what they wrote. You have to follow up and fix your paper based on these.
Yes.
But now there is scrutiny to the reviewers as well. Okay.
Okay.
The biggest change we are making now is that we will no longer accept reject decision following peer review, nor obviously ask our reviewers to make publishing recommendations. So again, this is 2 days ago. This is very significant. There will be a continuation to this story. Although we do not have the capacity to review every preprint submitted to us, we will publish every paper we send out for review as reviewed preprint, a journal style. Paper containing the author's manuscript, the e-life assessment, and the individual public peer reviews. The authors will also be able to include a response to the reviews and assessments. So basically, they are going to set up a blog for every paper. So what they are saying is the following. If you send us a paper to e-life, our editorial board will decide if it's acceptable topic and methods and whatever for our journal or not. If it is, we guarantee that it's going to be published a journal style. It will have a DOI. It will look, like a published paper. We will publish the reviews. We will publish comments from the public, and we will publish authors' response to reviewers or response to public. All of that. And now let the community decide what is the scientific value of this piece of work. So again, as I told you, we are living through an era where things are changing and changing quite quickly. What we used to think when I was trained as a scientist, peer review was sacred. And now, because of lots of problems, and we'll discuss these problems, things are shifting and changing. Any questions about the peer review? All right, so let's move on to the failure of the scientific method. We discussed before what the scientific method is. And the big failure is something that we are mostly dealing and discussing over the last 2 decades. It really starts with one revolutionary guy, one person, an MD, a doctor. He publishes in the year 2005 an essay which he titles, "Why Most Published Research Findings Are False." So this is intentionally a provocative title for a paper. His name is John Ioannidis. And he says in the summary of this essay, there is increasing concern that most current published research findings are false. Okay? He's one guy, 2005, and he keeps doing this. He writes several different comments. Here is, this is already just a public newspaper at the Atlantic. And this guy Friedman writes about Ioannidis, lies, damn lies, and medicals. Much of what medical researchers conclude in their studies is misleading, exaggerated, or flat out wrong. So why are doctors, to a striking extent, still drawing upon misinformation in their everyday practice? Dr. John Ioannidis has spent his career challenging his peers by exposing their bad science. So this guy is Don Quixote, and he keeps on doing this starting around 2005, and he talks about this in conferences and writes essays and papers and trying to prove that medical science, psychological science, and science in general, there are a lot of things which are wrong about it. And he points out what is wrong. Initially, it's being dismissed and ignored. Another paper from 2012. Why science is not necessarily self-correcting. History suggests that major catastrophes in scientific credibility are unfortunately possible, and the argument that it is obvious that progress is made is weak. We discussed the scientific method. We thought that, this is how we move forward the human front of knowledge. But there can be many accidents and even frauds and mistakes and wrong concepts. And basically, Ioannidis says, we don't have really means to correct the way we do science. And sometimes mistakes stay there for 30 years and somebody discover and reinvent the whole field because there was a wrong assumption and nobody really noticed. Efficient and unbiased replication mechanisms are essential for maintaining high level of scientific credibility. Basically, you need to repeat the results many times in different labs. In the absence of replication efforts, one is left with unconfirmed discoveries and unchallenged fallacies. In several fields of investigation, including many areas of psychological science, perpetuated and unchallenged fallacies may comprise the majority of the circulating evidence. He's even going as far to say that there are some fields that are based on wrong ideas, the whole field. Another paper from the same guy, 2015. Failure to replicate sound the alarm. And 2016, why most clinical research is not useful. These are, again, provocative titles, but he has done this by now for 11 years. And slowly but surely there is some traction and people start to listen. So far as to claim that cancer research is totally broken, and there is a committee, and there are conferences, and eventually National Institute of Health, NIH, in the US decides that one should look into replication of very famous experiments that the whole cancer research field is based on. They give funding to do those replication experiments. They choose 50 most notable experimental results that the whole cancer research is based on over decades, and ask people, let's replicate what, all our knowledge and all our therapeutic efforts are going towards the, this signaling pathway or this chemotherapeutical procedures and so on and so on. And the question is, all these research and billions of dollars that went over the years to do cancer research in this way or that way, if you go to the original papers, can we reproduce them? NIH decided that this is an important enough question, the replication crisis, and they're going to put money towards replicating results. Reproducibility in biology. So eLife, as part of this, and I told you about this revolutionary role of eLife as a new type of a journal for open science and now for new way of doing reviews, pick up that challenge. And eventually you'll see that the results of this NIH effort are going to be published in eLife. Reproducibility in cancer biology, the challenge of replication, interpreting the first results from the reproducibility project. NIH defined that project. It was called the reproducibility project. And here in 2017, the very initial results come. Cancer biology requires a highly nuanced approach. The aim of the reproducibility project, Cancer Biology, which is a collaboration between the Center of Open Science and Science Exchange, is twofold: to provide evidence about reproducibility in preclinical cancer research, and to identify the factors that influence reproducibility more generally. So that's a goal of the money that the NIH put forward. 2017, reproducibility project yields muddy results. That's already not a good sign. Ambitious effort to replicate cancer studies is provoking controversy. 2017, the reproducibility crisis. Reaction to replication crisis should not stifle innovation. The assertion that a 50% level of reproducibility equates to a crisis, or that many of the original studies were really fruitless, has been disputed by some specialists in replication. So obviously there is some resistance to this idea. And some people say, no, we don't have any crisis. We should continue to do the work that we are doing now, the way we are working now, and everything is good. 2018, plan to replicate 50 high-impact cancer papers shrinks to just 18. So as they set up the whole parameters, how this project that is funded by NIH is going to happen, initially they targeted 50 papers, very, very important papers in cancer research. And they said, we are going to reproduce those 50 papers. And already in 2018, they say, well, we cannot do this. For all kinds of reasons, we can try to replicate only 18 papers, not 50. And here are the results. This is just last year, December 7, 2021. So here are two papers published by the group of Arrington, who is in this Center for Open Science, funded by NIH. They assigned all those replication efforts. And there are two papers back-to-back in eLife, Reproducibility in Cancer Biology, Challenges for Assessing Replicability in Preclinical Cancer Biology. So let me quickly read the abstract for this first paper. We conducted the reproducibility project Cancer Biology to investigate the replicability of preclinical research in cancer biology. The initial aim of the project was to repeat 193 experiments from 53 high-impact papers, using an approach in which the experimental protocols and plans for data analysis had to be peer-reviewed and accepted for publication before experimental work could begin. So this is already referring to what I said, papers that are reviewed actually before the work is done. So basically you list only the plan of how you're going to do it, and that's what is peer-reviewed. However, the various barriers and challenges we encountered while designing and conducting the experiments meant that we were only able to repeat 50 experiments from 23 papers. Here we report these barriers and challenges. First, many original papers failed to report key descriptive and inferential statistics, the data needed to compute effect sizes and conduct power analysis was publicly accessible for just 4 of 193 experiments. The statistical analysis of the results in those main 53 papers was not good enough, and they could not really repeat the statistical analysis because there were no enough details given. Moreover, despite contacting the authors of the original papers, we were unable to obtain this data for 68% of the experiment. Second, none of the 193 experiments were described in sufficient detail in the original papers to enable us to design protocols to repeat the experiments. So we had to seek clarifications from the original authors. While authors were extremely or very helpful for 41% of experiments, they were minimally helpful for 9% of experiments, and not at all helpful or did not respond to us for 32% of the experiments. Third, once experimental work started, 67% of the peer-reviewed protocols required modification to complete the research, and just 4% of those modifications could be implemented. Cumulatively, these three factors limited the number of experiments that could be repeated. The experience drawn attention to basic fundamental concern about replication. It is hard to assess whether reported findings are credible. So this is really earth-shaking. They contacted the authors. The authors did not have good enough lab notebooks. The protocols were not listed properly. All kinds of information was missing, so they could not really repeat those experiments. And many authors, either died or moved on or whatever, didn't have lab notebooks, did not even bother to respond to their requests. So basically, they could not do the replication project properly because of all these issues. The second back-to-back paper from the same group of authors is titled Investigating the Replicability of Preclinical Cancer Biology. So that's a real paper. That is a paper that reports on the successful reproduced experiments. So a total of 50 experiments from 23 papers were repeated, generating data about the replicability of total of 158 effects. Most of the original effects were positive effects, with the rest being null effects. A majority of the original effect size were reported as numerical values, with the rest being reported as representative images. We employed 7 methods to assess replicability, and some of these methods were not suitable for all the effects in our sample. One method compared effect sizes. For positive effects, the median effect size in the replication were smaller than the original. So basically they say for the successful experiments where there was a mathematical number for an effect, the original reports overestimated the effect. We measure much smaller size effects. The other methods were binary. The replication was either a success or failure, and 5 of these methods could be used to assess both positive and null effects when effect size were reported as numerical value. For positive effects, 40% of replications succeeded according to 3 or more of these 5 methods. And for null effects, 80% of replications were successful. Combining positive and null effects, the success rate was 46%. A successful replication does not definitely confirm an original finding or its theoretical interpretation, so that's legal language. Equally, a failure to replicate does not disconfirm a finding, but it does suggest that additional investigation is needed to establish the reliability. So basically, they said we could not replicate most of the results. The result that we could replicate, it was only about 40% that could be replicated, and the size of the effect was smaller. So this is after 5 years of work, lots of millions of dollars from NIH, trying to repeat the most important and most high impact results in cancer research. And those cannot be replicated. So this is quite earth-shaking. Following week, obviously all scientific literature is full of commentaries. I won't go much through this, but all the editors of all important journals basically responded to these 2 papers. Key cancer results fail to be reproduced. Understanding the real reasons reproducibility reform fails. Disappointing numbers. Lack of rigorous is often blamed on pressure to publish, but ethnographers can find out what truly keeps science from upping its game. So this is really concerning. We talked about the scientific method. We trust the scientific method. We trust the peer review. And here you see that we cannot even reproduce what we published 2 decades ago. Barriers to reproducing preclinical results included unhelpful author communications. A high-profile replication study in cancer biology has had disappointing results. Scientists must redouble their efforts to find out why. So that's just cancer, but you can imagine that this is everywhere. I don't know how, so you're all physicists. Last year when I taught this course, there were quite a few people from life sciences as well. You're probably not that familiar with methods in life sciences, but basically everybody is using cultured cells, basically to the immortal cell that you keep happy inside incubators, and you can do biological studies on these cells. You have to feed cells that are cultured. You feed them with basically blood taken from the slaughterhouse, from cows. It's called fetal bovine serum, and they come in batches. You can imagine that there is a large variability in serum, and all the experiments done on cultured cells are now suspected because you are getting different results for different batches of different serums. So this whole crisis of replication, you will continue to hear about this. This is really huge issue, and it's not only in biology. Think about artificial intelligence, very popular nowadays. So AI itself is facing a big problem of reproducibility. Unpublished code and sensitivity to training conditions make many claims hard to verify. A computational challenge dares scientists to revive and run their own decades-old code. So as you move forward, operating systems are upgraded, hardware is being upgraded, code that you wrote 20 years ago cannot run on machine nowadays, and you have to rewrite the code in Python or whatever. And there is a huge problem there too. Is AI fueling a reproducibility crisis in science? So we talked about biology, we talked about computer science, and now it gets to AI, and it's all over the place. So let's summarize this chapter on replication crisis. The replication crisis is, as of 2020, an ongoing methodological crisis in which it has been found that many scientific studies are difficult or impossible to replicate or reproduce. The replication crisis affects the social sciences and medicine most severely. The crisis has long-standing roots. The phrase was coined in the early 2010s as part of growing awareness of the problem. The replication crisis represents an important body of research in the field of meta-science. So as I mentioned, because of this type of issues, the whole concept of the scientific method of the peer review replication crisis and responses to these problems is now packaged into a new scientific discipline called meta-science, which is the science of science. You use scientific method to study the way we do science. Because of reproducibility of experimental results is an essential part of the scientific method, the inability to replicate the studies of others has potentially grave consequences for many fields of science in which significant theories are grounded on reproducible experimental work. The replication crisis has been particularly widely discussed in the field of psychology and in medicine, where a number of efforts have been made to reinvestigate classical results to determine both the reliability of the results and, if found to be unreliable, the reasons for the failure to replicate. So what do you think are the causes for the replication crisis?
maybe not giving enough information about what you did in your experiment?
Definitely, that's part of it. I know you're all physicists and you think that physics is the most pure of all sciences and it couldn't happen in physics. I will convince you otherwise, but it's all over the place. And there are many reasons. The reason that Chaim brought up is definitely one of them. Non-professional biases. What do you mean by that? You can open your mic. Bias results.
I mean, professional, stuff like self-promotion, being put...
So basically non-scientific biases that somehow affect the whole process.
Yes, whether it comes to helping a friend out to benefit his name or promoting your own career, stuff like that.
Inconsistent scientific methods, more. So the causes for the replication crisis, publication and the selection bias. So we talked a little bit about this in terms of science and nature pushing for all kinds of stuff that really affects people's careers. Generation of new data publication at an unprecedented rate. Science has basically experienced exponential growth over the years. We are producing more than ever PhDs, and we build buildings in universities, and we hire more people. And just think about what is happening in China and in India, and countries that traditionally were not very strong in science are now getting much stronger and build. And basically, we have lots of journals popping up, all new journals popping up all the time, and we are generating scientific information in unprecedented rate. Obviously, a lot of it is not good, and we don't have all the checks and balances. Majority of these discoveries will not stand the test of time. Failure to adhere to good scientific practice and desperation to publish or perish, because this is how you are being promoted. When China about 10, 15 years ago decided to really push very hard to up their science, they were paying their professors for each science and nature paper that they were able to publish. And they stopped this practice because very quickly they ran into issues of misconduct, and they admitted this. But if the main motivation to publish is to get a position, to get a higher salary and so on, obviously the pureness of the scientific method is being contaminated. And this could drive, to wrong practices, wrong results, and that cannot be reproduced. Multiple various stakeholders. All right, so there are many, many issues, and it's obvious that we need to change to get science better. So what are the responses? As I mentioned already, the establishment of a new discipline called meta-science, that basically the hope is that once you research and you understand what are the reasons for doing science not in an optimal way, you can come up with better ways to do science. So that's what this new discipline promises to do. Tackling publication bias with pre-registration of studies, so basically changing the peer review and the way we publish papers. Shift to a complex system paradigm, emphasizing replication attempts in teaching. That's what we are doing today. Reducing the p-value required for claiming significance of new results. So many, many people have been using statistics in the wrong way, because biologists and psychologists oftentimes are not trained in statistics, and they still use it, and because they don't know enough, they don't use it properly. So it has been identified that statistical training is one of the biggest issues. Addressing the misinterpretation of p-values, again, relating to statistics. Encouraging larger sample sizes. Sharing raw data in online repositories. So now you know that all respectable journals require you not only to put your data, your figures and so on, but put the raw data in the repositories, put the code you are using for analysis in repositories. So this is all going under the title of open science. Funding for application studies, that's what NIH did for cancer. Emphasize triangulation, not just replication. So replication by itself is not enough. Get, several different types of methods to look at the same problem, take several different experiments and come to the conclusion from more orthogonal type of investigations. Raise overall standards of method presentation. And in general, what we call nowadays open science, which means open access. Journals used to require subscription. All libraries in all universities used to pay those fees. Individuals who belong to societies also paid subscription fees. But now, because all the funding for science, most of the funding for science comes from governments, from tax money. The idea is that anybody who pays taxes should be able to access scientific literature. That means that papers should be available for free for everybody to read. So that totally shifted the way publishers work. They really oppose this very, very strongly. And eventually the solution is that all journals have also a version of an open science journal where the cost of publishing, instead of coming from the subscribers, it's going to the authors. So the authors have to pay fees to get the paper published, but the papers are available for everybody. The raw data has to be publicly available. The analysis code has to be publicly available. There are some journals that are now asking your electronic laboratory notebooks. So the part of the problem why the replication project failed was that there were no good lab notebooks of those experiments. So now there's some journals that are even asking for your notebooks. So the bottom line, the idea is that in order to do better science, we have to give every piece of information so that somebody else can take all this information and try to replicate. All right, so this is what I prepared for today. Let me unshare. And if there are any comments, I would love to hear.
Actually, I have a kind of a question. Well, it's not related to topics that we studied today. It's like I'm Indian actually. So, well, most of the faculty members are the people who study at the premier institution, go to for the postdocs at the premier institution. When they go back again to the India, the work they did, the work they do, usually they are not cited that much. There's some kind of a biasing with the publication. So usually what they try to do is not publicly put the paper on archive first, send directly to the journals. And when the paper is up, means the reviewer or editor say that, okay, it will be published, then only they put it on the archive. So what's the...
Yeah, what you're saying is that there are some biases, geographical biases, that also need to be addressed.
Yeah, but so will that kind of means this open that e-life that you mentioned, will it overcome, will it help with this kind of...
I cannot speak for e-life, but...
no, I mean any kind of open kind of journal justice.
What you're presenting specifically, double-blinded review is probably the right answer for this, where the reviewers don't know who wrote the papers and the authors don't know who reviewed the paper. This is, I guess, erasing geographical biases or institutional biases and so on, but not all journals are doing this. So there is no one answer fits all. Many journals do different things. My goal here today and during the whole course is just to represent problems with the way we do science. And you are introducing another problem. And there are many problems. And if you want to have, and if you all, you all experience COVID, and science actually as a discipline suffered quite a bit from COVID because there is a big public mistrust in science because of initial, screw-ups in the way COVID was handled. And different stories came out, and the public did not know whom to trust and how. So we are in that era where everything is open. We, as scientists ourselves, claim that there are some problems, but the public also claims that there are problems. And we all need to get to do things better. So the purpose of this course is just to get you to think about this. I'm not giving really solutions. More comments? Okay, if no more comments, we'll end here for today, and we'll meet you next Tuesday. So good night, everybody.
Okay, thank you.
Can
Good night.
I ask something after the lecture?
So I'll stay online. Whoever wants to stay, sure. Go ahead.
All right, thank you.
Thank you.
Actually, earlier my microphone was not working. Will there be lecture next week?
Yes.
It will be there, like I think you mentioned one of the days it will not be there. So I
Yes,
could not know.
it's January 3rd, I think.
Oh, okay. 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 Thank you. Thank you.